Influenza Vaccine Effectiveness, Group 43, Project C, Paper 4, by Lauren Pitts, Brad Hamza, Maddie Young, and Casey Henneman. The paper being examined in this report is Low 2012 to 2013 Influenza Vaccine Effectiveness associated with mutation in the egg-adapted H3N2 vaccine strain, not antigenic drift in circulating viruses.
In 2012 to 2013, there was an early and intense peak of the influenza virus in Canada as well as the rest of North America. Influenza A-H3N2 subtype viruses were dominant and were associated with increased outbreak reports and showing a disappointing vaccine effectiveness of 45% for that component. The vaccine effectiveness was also as low in the US and Europe as it was in Canada. In order to find more about how to increase this vaccine effectiveness, tests were done. To test the end-of-season VE, Canada has since set up a routine centennial surveillance network used to estimate effectiveness of the annually reformulated trivalent influenza vaccine, or TIV. These participating centennial sites can offer nasal swabs for influenza virus to test all patients within 7 days of influenza-like illnesses. Using these test results and epidemiologic information, you could then calculate the VE for that particular flu season. Why such a low VE? The results were found that low VE for the H3N2 component during the 2012-13 season was related to mutations in the egg-adapted IVR 165 vaccine strain, rather than antigenic drift in circulating viruses. Realizing this, different strains of immunizations can then start to be tested, knowing that the 2012-13 flu season was related to mutations in the egg-adapted IVR 165 strain, rather than antigenic drift in circulating viruses.
This vaccine efficiency study is extremely relevant to the world of medicine. The topics discussed in this paper will have lasting impacts on the way we treat common illnesses like the flu. But these topics are also very relatable to our own Bio 1510 class. Themes like evolution, mutation, genetic drift, genotype, and phenotype are all prevalent throughout this paper. Let's take a closer look on how this influential research relates to our own course. Vaccine efficiency is historically related to evolutionary drift. Mismatch between the vaccine and the circulating strains was used to explain reduced protection among vaccinated individuals. This mismatch often results from evolutionary drift, one of the basic mechanisms of evolution. Evolutionary drift is used to explain the random fluctuations of allele frequencies among different populations. This mechanism of evolution is very common after a population bottleneck, in which the population decreases in size or diversity. Genetic drift can cause a population to become genetically distinct from the original population. This occurs due to the loss of rare genes and the decrease in the size of the gene pool. Genotypic and phenotypic characterization were the methods used to determine the true cause of the vaccine inefficiency. By closely examining the genotype and phenotype of the H3N2 strain of the influenza virus, researchers were able to determine that the true cause of low vaccine effectiveness was due to the mutations found in the egg-adapted H3N2 vaccine strain, and not from adaptations of the virus, as they originally thought. Having knowledge of why this vaccine was not working properly allows researchers to develop a more effective vaccine for future use. Influenza viruses that are capable of starting pandemics are injected into eggs to grow and develop. Once the virus is fully developed, it is harvested and used to create the newest round of flu vaccines. However, this study shows that instead of the virus mutating once it is spread among the population, it is the virus that is developing inside the egg to be used for vaccines that is undergoing the mutation.
Before we move on to the figures, let's check out a couple of definitions. Egg adapted. It means a virus that's grown on an embryonated hen's egg. And then antigenic drift is a mechanism for variation in viruses that involves the accumulation of mutations within the genes that code for antibody binding sites. And we'll see an example of that in just a moment. This figure illustrates the maximum likelihood phylogeny of the 57 sentinel viruses in the context of globally isolated viruses and recent vaccine components based on nucleotide alignment of the hemagglutinin domains. Vaccine components in previously reported clades are labeled and centennial viruses are colored by province. These three-dimensional structures we constructed using the crystal structure of an amino acid. Antigenic regions are color-coded purple, blue, light blue, green, and light green. The mutations between the circulation form and the virus are shown in red. This is the front view, and the left side is the circulation form of the virus, where the right side is the egg-adapted form of the virus, showing the mutations in red. The receptor binding site is labeled RBS. Notice how close the mutations are to the RBS. This proximity illustrates why the vaccine developed for the egg-adapted form won't fit the circulation form. This is the side view of the same virus, and the mutation is again shown in red.
This experiment was conducted in the 2012-2013 flu season, which started in November. Out of the 1,501 participants who were mainly 20 to 49 years old, 16% of cases and 26% of controls were immune to the flu virus. Most of the individuals had received the flu vaccine before, and it was found that if individuals had received the flu vaccination for 2 consecutive seasons, they were better protected. Ultimately, the results of this experiment show that the vaccine needs to be monitored for mutations along with the actual virus. The virus will change annually, but the vaccines might also need to be changed as well.